We've all heard of DNA, the double helix structure in Watson and Crick. In fact, we often give most, if not all, of the credit of discovering DNA to American biologist James Watson and English physicist Francis Crick. This, however, isn't necessarily the case, as in the late 1860s, DNA was first discovered by Swiss chemist Friedrich Miescher. His discovery, along with the work of other scientists as Phoebus Levene and Erwin Chargaff, carried out a series of research efforts that revealed additional details about the DNA molecule, including its primary chemical components and the way in which they are joined with one another. Without crucial discoveries and observations noted by these scientists, Watson and Crick may have never been able to make their groundbreaking discoveries on three-dimensional double helix structure of DNA. Let's take a look at some of the monumental breakthroughs surrounding DNA that have shaped the world as we know it today.
1869 was the year in which Swiss physiological chemist Friedrich Miescher first identified what he called nuclein inside the nuclei of human white blood cells. This term was later changed to nucleic acid and then changed again to deoxyribose nucleic acid, or DNA. Miescher's plan was to isolate the protein component of white blood cells, so he made arrangements from a local clinic to give him used bandages. He, however, came across a substance that had properties unlike any proteins, and therefore he concluded that he had discovered a new substance. In his journals, Miescher is quoted saying, "It seems probable to me that a whole family of such slightly varying phosphorus-containing substances will appear, as a group of nucleons, equivalent to proteins." Interestingly enough, Miescher never really received any press coverage or appreciation for nearly decades.
Following Miescher, other scientists continued to investigate the molecule formerly known as nuclein. Among these scientists was Russian biochemist Phoebus Levene, who was the first to discover the order of the three major components of a single nucleotide: the sugar, base, and phosphate. In addition, he was the first to discover the carbohydrate component, RNA, which contains ribose, and the first to discover the carbohydrate component of DNA, deoxyribose. He was also the first to correctly identify the way RNA and DNA molecules are put together. There were numerous alternate ways that the components could combine. Several scientists put forth suggestions for how this might occur, but it was Levene's polynucleotide model that proved to be the correct one. Based upon years of work using hydrolysis to break down and analyze yeast nucleic acids, Levene proposed that nucleic acids were composed of a series of nucleotides, and that each nucleotide was in turn composed of 1 and 4 nitrogen-containing bases, a sugar molecule and a phosphate group.
As time continued, many new facts and evidence emerged that altered Levene's proposals. Levene proposed that nucleotides were always in the same order, but scientists soon discovered that this was not true. Erwin Chargaff was one of a handful of scientists who expanded on Levene's work. As his first step in his search, Chargaff set out to see whether there were many differences in DNA among different species. After developing a new paper chromatography method for separating and identifying small amounts of organic material, Chargaff reached two major conclusions. First, he noted that the nucleotide composition of DNA varies among species. In other words, the same nucleotides do not repeat in the same order, as proposed by Levene. Second, Chargaff concluded that almost all DNA, no matter what organism or tissue type it comes from, maintains certain properties, even as its composition varies. The total amount of purines, A plus G, and the total amount of pyrimidines, C plus T, are usually nearly equal. But Chargaff himself could not explain these relationships, specifically that A bound to T and C bound to G within the molecular structure of DNA.
Using cardboard cutouts representing the individual chemical components of the 4 bases and other nucleotide subunits, Watson and Crick shifted molecules around on their desktops like putting together a puzzle. With the help of American scientist Jerry Donohue, the duo decided to make cutouts of the 2 bases. This results in the base fitting together perfectly, held together with hydrogen bonds while also staying true to Chargaff's rule. While there have been minor changes to the Watson and Crick model since then, the model's 4 major components have always remained true. They are
DNA is a double helix with two strands connected by hydrogen bonds. A bases are always paired with T's, and C's are always with G's. Secondly, most DNA double helixes are right-handed. Only one type of DNA, called Z-DNA, is left-handed in shape. Thirdly, DNA double helix is antiparallel, which means that the 5' end of one strand is always paired with the 3' end of its complementary strand, and vice versa. Lastly, the outer edges of the nitrogen-containing bases are exposed and available for all potential hydrogen bonding as well. These hydrogen bonds provide easy access to the DNA for other molecules. Of course, Watson and Crick played a vital role in discovering the structure of DNA, and their legacy will continue to shape the way that we understand science down to its rudimentary form. There is, however, much credit due to the scientists who preceded them, and due to that, it is important to recognize their discoveries and influence on science as we know it today.
As recently as last month, knowledge about DNA was applied in a study about Egyptian mummies. Using DNA from mummies, scientists are gaining deeper insight into the history of ancient Egypt. Scientists have said they are examining genome data from 90 mummies, making it the largest genetic study ever conducted about Egyptians. The DNA was extracted from the teeth and bones of mummies from as far back as 1388 BC to 426 AD. The scientists are answering questions like, are modern Egyptians direct descendants of ancient Egyptians? Was there genetic continuity in Egypt through time? Did foreign invaders change the genetic makeup? The genomes show that unlike modern Egyptians, ancient Egyptians were not very associated with sub-Saharan populations. The closest genetic ties were to the people of the Middle East. This is an example of just one of the amazing things that an understanding of DNA can do.